Hmm. The background is now working, but not working well. Sorry for being late here. My computer is really, really slow, so be with me. Bear with me. The Zoom meeting from the lecture is still going. We're at, well, I guess, at 69%, so just bear with me. Let me share my screen here. There we go. Can somebody please confirm you can hear me?
I can hear you.
Thank you. Oh, what's that? Let's close that down. Let's go. All right, before we begin, are there any questions? Let me repeat, this is part of the same course as the lecture, so I'm not going to talk much about the syllabus, but I do need to talk a little bit specifically regarding the lab. The same late assignments hold. Gosh, what's happening with my computer? It's not working well because so much energy is being spent on converting the lecture class into a Zoom movie so we can post it online. And all right, the grading of the lab will count towards the grading of the course. Um, you're not going to be using real microscopes, but we do have a virtual microscope for you to use in the lab, and you do need to learn how to use a real microscope and care for a real microscope, even though you won't be using one in the class. Safety information for students and physicians. All of our lab is going to be done online, so you should not be exposed to any microbes from the microbiology lab. It is important that you know this information, though, because as future nurses, dental hygienists, or healthcare workers, it's important to be aware of these safety issues concerning microbes. You will not become infected with the microorganism or COVID-19 from other students, but you need to know about them because this is a microbiology class. And so we need to teach you about safety and microorganisms. The microbiology labs, at least at Clark College, are operated at a biosafety level 2 level, also called BSL 2, where we wear protective equipment such as lab coats, gloves, goggles, etc., required for all work in the lab. It's true the goggles don't need to be always worn, but whenever you have a splash hazard, you have to use goggles. Access to the laboratory is restricted. Students who are immunocompromised, living with or caring for an immunocompromised individual, are pregnant or may become pregnant, or caring or live with someone caring for infants, should consult with their physician to determine the appropriate level of participation in the lab. Because we're doing this online, you don't need to know or be concerned, but you normally would. And then come and let me know. And like I said, we would give you a microorganism to work with that would be perfectly safe. In this lab, we grow microorganisms at biosafety level 2 or biosafety level 1. If you experience symptoms with any bacteria, you would want to contact your physician. Like I said, you won't be exposed to a microbe in the online class. But we have these biosafety level 1 microorganisms in the class. Oops, didn't mean to do that. Ah, shoot, what the heck happened there? Like I said, my computer isn't working right. It's because so much energy is being spent to convert the there we go. We also have these BSL-2 medium risk organisms in the class. Any question about any of that? All right. Some lab safety rules. You're not really going to be in the lab, but you do need to know about lab safety rules. We're not going to be physically meeting in the Clark College lab, but safety is one of the main skills that um, employers want their students to learn. So we're going to learn about safety. Never take shortcuts, which risk the safety and accuracy of the microorganism, of you, and then the students around you. Develop a positive, respectful attitude towards microorganisms. Yep, finally finished. All right. That was the lecture class. It just finished. So hopefully my computer will start working right. You're not allowed to take personal items to your lab bench. They must remain in your book bag or your bag. And you put those in a space provided for bags. When you first walk into the door and then the place where you put your bags is considered outside the lab, at least the lab proper. And this is where you put your book bag, your coats, your books, your purse, whatever else. You put it in your cubicle or your cubby hole, and then the door. And then you walk a few feet, and that would be considered inside the lab proper. Any questions about any of that? So the door and where you're storing your personal items in the cubicle is considered outside the lab. You then walk over to the sink, go wash your hands, do it well for 20, 30 seconds everywhere, washing everywhere. And then you go and put on your lab coat. And obviously, while you're walking to the sink, or that's inside the lab, and then go put on your lab coat. We have lab coats for students to use in the lab. They're kept in the lab. You would use the same lab coat all term long. At the start of the term, they have been cleaned and autoclaved, so they're sterile. And then at the end of the lab, meaning the end of the term, the lab coats would be autoclaved and then washed and then made available for the students for next term. It is possible for you to buy a lab coat, in which case you bring it into the lab for the very first day. And then once it's in the lab, it stays in the lab all term long. And you would reuse it each time you come into the lab. And at the end of the term, if you want the lab coat back, if you bought it, you then go and grab it, give it to your instructor. You'll put it in a good place and turn it into the lab prep people, and they will autoclave it and then wash it, dry it, and then what they would do is put it outside the lab. In between terms, you could come and pick up your lab coat. Any question about any of that? So your lab coat stays in the lab. If you bought it, and obviously if you're using it from us, it stays in the lab. And it never leaves the lab until it's been sterilized. And if you bought it, you can then pick it up and take it home. Once you get your lab coat on, so you wash your hands, you go and put your lab coat on, and then you put on gloves. We have gloves for you to use in the lab. And that way, when you're touching microbes, you should never be exposed to the microbes because you take off the gloves at the end of the day. If you're working with a splash hazard, you then go and get goggles. We have goggles in the lab. And they're sterilized by UV light in between use. So we reuse the goggles. I guess we reuse the lab coats, but you'd be the only one using that lab coat in the term. With the goggles, there'd be other students using the goggles, but you'd be the only one using the goggles on that day. And like I said, they're sterilized with UV light. You only need to use goggles whenever there's a splash hazard or you're using a chemical, which if it gets in the eye, could damage the eye. And generally, there's only one chemical that I recall that we use, which is hazardous to your eye, and it's the one of the gram stains, the crystal violet. The splash hazard would be anything that is liquid, which has microorganisms in it. So if ever you're using a liquid culture with microbes in it, you'd have to wear the goggles. Once you're done with the splash hazard or with the chemical that could damage your eye, you could then take off the goggles and then continue working in the lab. Any question about any of that? All right. At the end of the lab, when you're all done and about to leave, you then would take off the gloves. And if you don't have microorganisms on them and you didn't spill anything with microorganisms on them, you could then take them off and put them in the regular trash can. You would then take off your lab coat, hang it up where we have the used lab coats in the lab, and then go walk to the sink and then wash your hands. And then you walk to where your books are, and that's considered outside the lab. Grab your things, then walk outside the door, and then go home. So as soon as you walk from the sink to where your books or cubbyhole is, that's considered outside the lab proper. And then you leave the door, so you obviously left the entire room. Any question about any of that? All right. Another thing you do before you ever work on your lab bench, before you begin work, is you would disinfect the area you're going to work with. So you have a work area, your workbench. You disinfect it with disinfectant and wipe it up with paper towels. And then you do that at the start of every lab day. And then before you leave, you disinfect your work area. So you kill all the microbes that may have fallen onto your workbench. And you do that before you leave, before you take off your gloves, before you take off your lab coat, before you go to the sink and wash your hands, and then before you pick up the books in the cubbyhole and then walk out the door. Any question about the procedures that we work or follow in the lab? So you do that every day. You come into the lab, you walk in the door, you put your bags in your book, your coat if you have it. Obviously, in the summertime, you won't have a coat in your cubbyhole. And then you walk a few steps and you walk into the lab proper. You then wash your hands, put on your gloves, put on the lab coat. And if you're working with a splash hazard, you put on your goggles. Most of the time, we will not be working with a splash hazard or a chemical that would damage the eyes. So most of the time, you would not put on goggles. And then you go to your workstation, your workbench, and you disinfect it. And then you do your work. And then at the end, you disinfect your work area again. Uh, Take off your gloves, take off your lab coat, go to the sink, wash your hands, and then walk towards the cubbyhole. And as you're doing it, you're now leaving the lab. Grab your things from the cubbyhole and walk out the door. Any question about that? Only enrolled microbiology staff and students are permitted in the lab unless authorized by the instructor. Anything that's a personal item that you take to your workstation, such as books, purses, cell phones, would be confiscated by your instructor, and you would not get it back until it had been sterilized or put into a bucket of disinfectant. That would destroy all cell phones. It may destroy your books, your purses, and your book bags. So don't take anything personal to your workstation. You will be provided with paper and pencil near your workstation. We give you a drawer to work in, and you can keep things in the drawer all term long. Um, and if you need to write something, you can write it on the paper that we provide. And then that stays home. If you want to take notes, what you do is we have I think it's an iPad, and you take a picture of your notes or all your notes, and then email it to yourself, and that's how you can get your notes. Any question about any of that? So nothing that comes into the lab ever leaves the lab proper. And you shouldn't take your personal items into the lab proper because you won't get it back until it has been either autoclaved or stuck in a bucket of disinfectant. So cell phone usage, if you hear your phone ringing, you would have to take off your gloves, take off your lab coat, go wash your hands, go to your phone, which probably would stop ringing by that time, walk out of the door of the lab with your phone, and then talk to the person. And you might have to call them back because it's been so long. Then you come back into the lab, put your phone away, go to the sink, wash your hands, put back on gloves, probably fresh gloves, and then put on your lab coat. Or I guess put on your lab coat and then put on the gloves. Any question about that? So those are required. You must wear closed-toed shoes while you're in the lab. If you're wearing sandals or thongs on your feet, or a woman's fancy shoe, which has an open toe, that is not allowed in the microbiology lab. We want your feet to be at least somewhat protected in case you spill something or drop a bottle containing a liquid culture or even a solid culture. We don't want your feet to be exposed to microorganisms. So you must wear closed-toed shoes while in the lab. Now you can wear uh, sandals or thongs on your feet and then bring your shoes and just change your shoes, put on the closed-toed shoes before you enter the lab proper. And then put your sandals in the cubbyhole. In the summertime, I sometimes have students doing that. Cleanliness is a virtue in the microbiology lab, so always follow this procedure. You should also note where fire extinguisher, fire blanket, eye wash station, and first aid materials are located. For this lab, all you need to know is we have these available for you in a real microbiology lab in case you needed to use them. Accidents should be reported to instructor as the following. Clean up broken glass first. Broken glass can cut your skin and is likely to be coated with numerous microorganisms. Always use a brush or a broom and a dustpan. Never pick up broken glass with your fingers. Dispose of the broken glass in the broken glass container receptacle. If you have strong acids or bases, which we almost never use, I would say we never use in the microbiology, you should use the what do you call it, an activating agent. It's probably sodium bicarbonate to weaken the strong acid or the bases. And spilled bacteria culture should be cleaned up as follows. Cover the spilled material with lots of paper towels. Drench the paper towels with table disinfectant. Let the disinfectant sit for 15 minutes before you clean it up. Meaning we want to make sure the microorganisms are killed, so expose it to 15 minutes of the disinfectant. Sweep up any broken glass and then wipe up the rest with dry paper towels and discard of this waste in the biohazard trash can. We have a biohazard trash can, which is separate from a regular garbage trash can. Any question about that? The biohazard trash can will be taken and autoclaved before it will be disposed of, put in the regular trash. The regular trash will not be autoclaved. It will be just taken away with the regular trash. Be sure you keep hair, paper away from Bunsen burners and back to incinerators. Long hair should be tied back. We do have hair ties in the lab if you don't have one, or you don't have a way to tie your hair. Back to incinerators, and Bunsen burners should be turned off before you leave the lab. Mouth pipetting is not allowed in the lab. Never take cultures out of the lab. Samples can only be brought into the lab after you've gotten clearance from your instructor to bring it into the lab, which almost never happens, but I once had a student testing, what was it, herbs. She brought in herbs to test antimicrobial properties of the herbs or whatever, oils. If you have a cut or a sore on your hand, you should wash your hands as normal. Go over to our first aid kit and apply a bandage to the cut and then put on a glove. And then wash your hands at the end of the lab, and you can grab a Band-Aid if you want a fresh Band-Aid after you wash your hands. All microbial cultures must be labeled completely in the lab with your name, the date, the nature of the specimen, meaning what it is, and the medium that it is, meaning the medium that you're using. All specimens must be discarded properly in the following ways. An agar plate, which is in a plastic dish, is discarded in the biohazard container. All microorganisms on paper towels or something that's plastic should be put in the biohazard container when you're discarding them. If it's on something that's glass, glass should never be put in the biohazard container. Glass slides should be put in the container for glass slides. Glass cover slips should be put in the container for glass cover slips. What it is a dish containing disinfectant. And then cotton swab should be put in the cotton swab container. Once again, it's a dish with disinfectant. You can find the glass slide and the cover slip, glass cover slip container in the back of the lab, and the cotton swab containers are found in the back of the lab, and there's one, at least one in every table as well. Pipettes should be put in the appropriate labeled container. If they're plastic and not contaminated, they go in the regular trash can. If they're plastic and they've been exposed to bacteria, they go in the autoclave, the biohazard container. If they're glass, they go in where glass pipettes are labeled, and that would be in the back of the lab. Glass test tubes, which, well, it doesn't matter if they're contaminated or not, they go in the glass test tube container, which is in the back of the lab. All tape should be removed from the test tube, and what will be happen is that the glass test tubes will be autoclaved, and then they'll be washed, cleaned, dried, and then made available for further student use. The glass swabs, they're actually sterilized or close to sterilized in the disinfectant, and they'll be taken out of the disinfectant and washed, dried, and then made available for student use. The cover slips we do not reuse, and they will be put into the glass cover slip container and they'll just be discarded in the broken glass container. Supplies for the lab will be in the current lab will be used in the front of the lab. There will be some continuous supplies in the back of the lab, meaning bottles of media and stuff like that. Each pair of students are assigned a drawer uh, underneath their lab bench, their work area, and they will be the only students using that drawer for the term. So everything that's in the lab stays in the lab and never leaves the lab. Remember, where you keep your bags, your book bag, your books, your coat, whatever, is considered outside the lab proper. You're required to follow these procedures, otherwise you'll jeopardize the safety of others, and you could be excluded from the class if you didn't follow it. This is an online lab, so I'm not terribly worried about it. All right, so in the schedule for today, we're going to do Lab 00 today, going over lab safety. You must turn in this worksheet by 8 a.m. Friday. And on Thursday, we'll do Lab 01, Ubiquity of Microorganisms. It also must be due by 8 a.m. Friday. Remember, the environmental quiz is due this Thursday by 11:59 p.m. And then the plagiarism tutorial and the plagiarism quiz is due by 11:59 p.m. this Saturday. Any question about any of that? All right. Let's begin Lab 00, Lab Safety Training. I think that's the one I want. Maybe not. I'll have to make do because I don't have my highlighting in here. Yep, all right. All right, let me get the worksheet for Lab 00.
I just have a question. When I'm trying to open the lab on Canvas, I don't know if it's not showing up correctly because I need to have Microsoft 365 downloaded. Is that why maybe mine looks weird?
I'm not sure. Let me take a look. Computer is still kind of slow. I don't know why that's the case. Have you downloaded it?
Yeah, I
Because that would
tried.
help rather than using in Canvas.
Yeah, the way
It
it's
looks something weirder than this.
yeah, like it's showing up like what is it called? Like a script. It's not showing like any type of document. I'm gonna try to download my Microsoft on here. I have a new computer, so I just need to download it real quick and see if that might help.
Okay, I'm not sure what the problem is. You can use Microsoft directly from Clark College. Um, Probably the easiest way to do it is just log into, see if I still have it here. I don't think I have it here anymore. Yeah, I don't have it here anymore. But you can use Microsoft by um, just logging in as a Clark student, and then it'll give you the option of what you want to open, and you just say Microsoft Word. All right, let's go on to the lab. I got lots of these open, I think, now. It looks a little different than this one. Looks like you got this one. All right. Anyone working in an unsafe manner in the lab can be banned from working in the lab. Obviously, this is an online class, so it won't be, you won't be banned from the online class. You're not going to be working in the lab, but you need to know these materials. Many of these rules are relevant during your career. The learning objectives of this lab is be able to distinguish between safe and unsafe laboratory practices, be able to understand where relevant safety equipment is located in the lab, understand how to safely dispose of contaminated materials. You should always wear proper attire while in the lab. You must wear a clean lab coat that is buttoned, covers your lap when sitting. You must be wearing disposable gloves. You must have closed-toed shoes. And if you're working with a splash hazard or a chemical that can damage your eyes, you must wear goggles. Now note, most of the time you would not be wearing goggles in the regular Clark microbiology lab, because most of the time we would not be working with a splash hazard or a chemical that could damage your eye. Long hair should be tied back. Absolutely no food or drink at any time may be eaten or used in the lab. You may not use electronics. Anything that's brought into the lab will be confiscated from you, and you will not get it back until it has been cleaned in a bucket of disinfectant or autoclaved, and that would destroy a cell phone. Absolutely no personal items allowed into the lab. All personal items are stored in the storage area, the cubbyhole, and this is considered outside the lab, although it's inside the door. And I've already mentioned that. So we'll skip that. This is what you should look like while you're in the lab, where you have closed-toed shoes. You should have a lab coat on. You should have gloves. And I can't tell if that's goggles or the person's glasses. If you're wearing with splash hazard, you should have goggles. If you're not wearing a splash hazard, you do not need goggles. And those could be the person's glasses. And then the person has a tie on their hair because they have long hair, which you can't see. And this picture actually tells you some information about the worksheet. It says what must be worn for safety before you come into the lab. A lab coat is something you put on once you come into the lab. So a lab coat is not something you wear before you come into the lab. The shoes would be something you put on before you come into the lab. The pants, you can't see it, but maybe a little bit of the blouse. Those are things you're putting on before you come into the lab. So that picture helps. Keep your work area clean and uncluttered. You should disinfect the lab bench before you begin your work in the lab, and then at the end of each lab session. That's actually a picture of our microbiology lab right there. And so this region here should be disinfected before you begin and then after you leave or about to leave. Discard of waste in the appropriate receptacles. Regular trash should have no sharp objects, no glass, and nothing contaminated with bacteria. So that would be a regular trash can, which is not shown here. A biohazard container is shown here. You put contaminated material with no glass and no sharps. We don't really use sharps in microbiology like a razor blade, but you'd put no sharps and no glass in the biohazard container. But anything that's plastic that's contaminated with bacteria or anything paper, like a petri dish, it's plastic, goes in the biohazard container. It will be autoclaved. You can see that this plastic bag can be taken out, can be put in the autoclave, sterilized, and then it would be sent in Clark College regular trash. Any question about any of that? Contaminated test tubes, anything glass, which we don't really use other glass containers other than test tubes, would be put in the back of the lab by the test tube container. And you should stand them up and remove any tape from the test tubes, meaning the test tube holders where you put them. We have different containers for bacterial swab, meaning cotton swab. That should go in the cotton swab container. It has a bucket of or beaker of bleach. We have one for slides and then one for cover slips. They're all different containers. They all contain disinfectant. You should consider all materials contaminated as potentially pathogenic organisms. If you're unsure how to dispose of something, ask your instructor. Report all spills and accidents immediately, no matter how slight. Cover the material with a paper towel, then saturate the area where the spill is with the disinfectant. Let it stand for 15 minutes before wiping it up with clean paper towels. The paper towels and the disinfectant should be put in the biohazard container. If it's glass, after you do this, you'd then put the broken glass in the broken glass receptacle. And separating the broken glass from the other waste would be a chore, but make sure you use a broom and a dust pan to collect the broken glass. Don't use your fingers. Keep lids on reagents when not in use. Never pour reagents back into a container. Once it's out of the container, just discard it. Don't put it back. Do not pour chemicals down the drain. We've got waste for hazardous chemicals, unless you get permission from your instructor. And then don't pour a chemical into the regular garbage trash can either. That should be solid waste. No mouth pipetting, no chewing your fingernails, no chewing a pencil while you're in the lab. Never leave a Bunsen burner or a Bacto incinerator on if it's not being used. Obviously, to get the Bacto incinerator on, it has to be turned on 5 minutes before you use it. So usually students, when they come into the lab, they clean their workbench, disinfect their workbench, and then they turn on their Bacto incinerator. 5 minutes later, it'll be red hot and then ready for use. They should be only put in the Bacto incinerator or the flame when you're sterilizing the tool. Meaning don't leave it in the Bacto incinerator and the flame. Be familiar with the location. You don't need to be familiar with the location of the safety equipment, but you should know we have an eye wash station, shown here. We have a fire extinguisher, shown here. We have a blanket, which might be shown there. We have a deluge shower, shown here, a first aid kit shown here, and a fire alarm. All of these safety equipment is in the lab, ready for your use should you need it. I've never had to have a student use the fire blanket or the chemical shower, meaning, but they're there in case it's needed. In fact, I've never had a student actually need the first aid kit, but we have that other than for putting on a Band-Aid. I've never had a student need that either. Fire alarm, never used. Eye wash station, I've never had a student use it. I once had a doctor, a Chinese doctor. This is before I was, actually when I was a graduate student. He got a chemical in his eye, and I took him over to the eye wash station and helped him wash out his eye. But that's the only time I've ever used the eye wash station. So hopefully we'd never use it again. But we have all of those available for you in the lab. Never use the fire extinguisher in the lab either. All right, you should pre-read the lab before you do the lab. When in doubt, ask a question. Obviously, you can't pre-read this lab because I just opened it up. The final 2 pages of the document detail the safety rules, which are already covered in the syllabus for Clark College. You are expected to follow these rules. So you can read over them again if you need to. there will be no safety deduction because there's no way to really do something unsafe online. You should consider all cultures you're working with to be potentially pathogenic. Remember, you have to use closed-toed shoes while in the lab, microbiology lab at Clark College. Never take cultures out of the lab. Everything that's in the lab stays in the lab. And don't bring things into the lab proper. Once it's brought into the lab proper, it either has to stay in the lab proper indefinitely, or at least until it's disinfected in a beaker of disinfectant or been autoclaved. And then it can leave the lab. All right, any questions about this lab? If not, let's go to the worksheet for lab 00. I'm not finding it now. There it is. Fill in the answers to each asterisk. So when I have 4 asterisks, that means there are 4 answers to rule 1. In the lab, what do you always wear for safety before you enter the lab? This is not directly stated in the lab, but it is shown in the picture in the lab. Okay, any questions about that? Rule 2. After you enter the lab, what 2 items do you put on to keep yourself safe in the lab? Now, the lab coat, is that something you put on to keep yourself safe after you enter the lab or before you enter the lab? Can anyone answer that?
After.
That's correct. All right. Rule 3, look up safety data sheet online. You have to do that online because the lab doesn't discuss it. Each chemical in the lab has a safety data sheet, or SDS, that describes what? Provide at least a phrase for each answer, meaning one-word answers are unacceptable, and you got to list 1, 2, 3, 4, 5 different items that the safety data sheet describes. Okay? Obviously, the chemical composition of the chemical you're working with will be one thing. Okay? Rule 4, name 5 items of safety equipment found in the lab. So each asterisk should have an answer. By safety equipment, we mean a piece of equipment. So goggles and gloves is not a safety equipment. Rule 5, name 2 items you can take into the lab. I didn't directly state it, but it is stated in the lab manual, and that is anything that you're wearing can come into the lab. So if you're wearing a necklace, or you're wearing an earring, or you're wearing a watch, you can bring it into the lab. Any questions about that? Now, for the watch, it should go underneath the glove, and you just should not touch your earrings while you're in the lab. But you can bring in anything that you are wearing. Obviously, your clothes, you're going to be bringing that into the lab as well. So there are items that can be brought into the lab. Then can you eat and drink in the lab? That's the third question to Rule 5. 2, list 1 item of laboratory attire you must wear in a microbiology lab. 3, list 1 item belonging to you that is allowed to be brought into a microbiology lab. And then 4, list 1 item belonging to you that is not allowed in a microbiology lab. And for rules 3 and 4, I am not talking about something like a lab coat, which belongs to you, and then you only bring it into the first lab. So that will not be accepted because it will not be allowed into the lab on the second lab day, or the third lab day, or the fourth lab day. It should only be brought into the lab the first lab day. So the lab coat would not be an acceptable answer. So this is 1 item that you can bring into the lab anytime in the lab. And then 5, submit this file by 8 a.m. Friday, week 1 in the folder for worksheet lab 00. And if you do not, as well as the worksheet for lab 01, you will be dropped from the class. I'm going to use these 2 assignments in the lab to record attendance for week 1. And somebody who's not attending the class will be dropped. Any question about any of that? All right, if not, go ahead
I
and
have
get
a question
started
on
the
number
lab.
1.
Uh-huh. Talk about rule 1 or number 1?
Sorry, rule 1.
Okay.
Rule 1, it says, what do you always wear for safety before you enter the lab? And it's referring to the picture,
Uh-huh.
but the lab coat and the gloves would be items after you enter. So, and it has 4 asterisks there. So I'm just kind of confused about what the 4 items are.
Well,
If
I'm
it's
not going to
referring
tell you the answers, but you should be able to
No, I'm
find
just confused.
in the
Is it
picture.
referring specifically to that picture?
The picture shows you 4 items. I'll tell you one thing. Your shoes, your closed-toed shoes,
Yeah.
you normally put on before you enter the lab. Right?
I guess I'm looking at the picture where it has the student standing there, her hair's back. Hair's pulled back and secured, closed-toed shoes. But then the other 2 arrows are pointing at gloves and the lab coat, and those wouldn't
No,
be
the
things.
arrows are not pointing at something you put on before you enter the lab.
Oh,
They're
okay.
not doing that. So if you find something in the picture.
Oh, okay, gotcha. I thought it was just referring to those. Okay.
Okay, all right. I've now lost that background again. It's a little bit right there. All right, so go ahead and work on the lab. I'll be here until 9 o'clock or the last student leaves the lab. Once the last student has left the lab, I will not be here to help you with any answers in the worksheet. But if you have a question, just ask. Like I said, I'll be here until the last student logs out or until 9 o'clock to help you with the lab.